Good morning, good afternoon, and good evening, everyone. My name is Jose Gonzalez. As Stacy stated, I have been with the technical support group here at Illumina for over 6 years, supporting the BeadArray product line. And today's topic will be Manifestology, deciphering Illumina BeadArray manifests. All right. Our objectives today are to understand what a manifest is, how GenomeStudio uses it to create its GenomeStudio projects, to review information contained in the various types of manifests for the various types of BeadChips, to understand strand designations and how these designations can be used to decipher the various output reports from GenomeStudio, and to review various additional resources available that supplement the information that is in the manifests. First, before we can understand what a manifest is, we should have a good understanding of what our BeadArray technology is and how it works, and how the manifests are involved in deciphering that information. So, starting with the BeadChip. A BeadChip is a silicon wafer which has pits in it into which beads that are coated with our oligos have been dropped. Probes are coated onto the beads, which are in turn distributed randomly over a large section of the bead chip for each sample. Now, each of these stripes, after the beads have been distributed randomly, gets mapped, and a descriptive file called a DMAP is generated for each stripe on the bead chip. A DMAP lists a coordinate in the stripe and the identifier of the bead type at that coordinate. All right. The bead type identifier is a number called an address. So essentially, a DMAP is a table with a list of coordinates and the corresponding bead identifier for the bead type that is occupying that coordinate. Now, bead types are represented multiple times per sample. As you can see in this diagram, there are multiple red beads, multiple yellow beads, and those colors simply represent particular bead types, which in turn are coded with each with a single particular type of oligo. All right. What happens when a scanner scans a bead chip? The scanner creates an image for each stripe. A grid is overlaid on top of that image, and the DMAP table is applied so that each bead on the image is identified properly. The scanner then extracts the intensity for each bead type from the image and outputs that information in what we call IDATs, intensity data files. There's an intensity data file for each stripe, for each channel that the scanner scans the stripe on. Our scanner can scan in the red channel and the green channel. So gene expression bead chips only scan in the green channel, so they only get one green IDAT per sample, whereas methylation and genotyping bead chips are scanned in both red and green, so each sample gets a red IDAT and a green IDAT. What's in an IDAT? The bead type information after scanning is consolidated. In other words, for each type of bead, all of the beads are averaged, and so the average is listed per bead identifier, or per bead type. For each bead type, the scanner removes outliers and then averages the remaining beads and lists it once, so that in an IDAT file, although each bead type is present on the stripe multiple times, in an IDAT, each bead type is listed only once, along with the average intensity, the number of beads used to calculate that average, and this bead standard error, which is the standard deviation of the average intensities. So that's what an IDAT file contains. Now, what is a manifest and how is it used? A manifest is, the bottom line, a list of the probes contained on a bead chip or oligo panel. The manifest contains the address codes used by GenomeStudio. These address codes correspond to the address codes in the IDATs. Intensity information is merged by GenomeStudio with the manifest in order to create a meaningful information regarding the intensities and the probes. So the manifest also may contain information that further defines the probes, things like gene annotations, genomic location of the probe or the probe target, information regarding the source of the information for the probe targets, strand designations, and probe sequences. So that's what's in the manifest. Now, once the IDATs and manifests have been pulled by GenomeStudio, GenomeStudio creates a GenomeStudio project. A GenomeStudio project lists the probe IDs and the corresponding addresses. It lists the intensities for each sample derived from those probes. It displays the secondary data that's calculated from those intensities, things such as genotypes, things that need to be derived from the intensities of the probes, methylation state calculations, things like that. And it also carries over the annotations and other information that's contained in the manifest. So this is, in a nutshell, what a GenomeStudio project is. The intensities of the probes, the secondary processed data, and the annotations. All right? Now that we know what a manifest is used for by GenomeStudio, let's take a look at some of the manifests. Before that, in particular for genotyping, we need to understand strand designation. This is because a SNP is a double-stranded phenomenon. And so you need to look at SNPs as double-stranded phenomena. When a SNP is reported in a database, it usually only refers to one strand or the other. And so we have to identify which strand the SNP is being reported on. Also, keeping in mind that when we design the SNP assay, we look at both strands so that and then choose the best one for the assay. And so we need to keep in mind always that the probe can be designed on either strand for the SNP. Now, Illumina invented a strand designation on its own. We invented the top/bot strand designation and also the A/B genotype designation. The reason we invented this was because when SNPs were first being registered into databases, there was not a complete reference genome that had been completed. And so if we tried to reference the strand being reported in the database to a genome strand, it would often switch back and forth as the genome was being completed. So we wanted to invent a strand designation that would never change, regardless of what changes would occur in the reference genome or in the databases. So we invented the top/bot designation and the AB genotype designation. All right. We do have a technical note that describes exactly how the top/bot and the AB genotype designations work. We also have a technical bulletin that was written by one of my colleagues in the technical support department, and it was recently released. Thankfully, it's getting a lot of hits because people want to understand this. So I do recommend that you use one or the other or both. All right. Now, in a nutshell, I've put how we define top and bot in one slide. First, we designate bases A and T as determinant bases because we use these bases to determine the strand and AB alleles. Now, for non-AT and non-CG Snips, that is AC, AG, TC, and TG Snips, A and T alleles are always considered genotype A, regardless of which strand you're looking at, when one is A and one is T. So when you have a case where A and T are on the two strands, then you have a genotype A case. C and G are considered genotype B. So for these non-AT and non-CG SNPs, it's relatively simple. The strand containing the A allele is called the top strand. The strand containing the T allele is the bot strand. So for non-AT and non-CG SNPs, it is relatively simple. For AT and CG SNPs, because you cannot use A and T genotypes or C and G genotypes to determine either top or bot strands, or A and B genotypes, we need to use the surrounding sequence to determine the strand first, and then assign which alleles A and T or T and A are genotypes A and B, respectively. So we use what we call a strand walking procedure to determine the strand and then assign the genotype. So we'll go through a few examples of that. For non-AT and non-CG SNPs, it is, again, relatively easy. It's quite simple. If you list the genotypes here, this will always be true. AG and AC genotype will always be top because of the A. TG and TC will always be bot. A's and T's are always going to be allele A, and C's and G's will always be allele B. Now, for non-AT and non-CG SNPs, I created a table here. You can try to fill it in if you want and quiz yourself, but the answers are all here already on the slide afterwards. So you can block these out and try it yourself to see if you understand it fully. All right. Now, for the AT and CG SNPs, as I said, we need to look at the surrounding sequence. So we do a strand walk. So first we look at the first base upstream and the first base downstream. We look for a case, an instance, where one of the bases is an A or a T, a determinate base, and one of them is not. And in this particular example, you can see that the T is on the 3' side of the SNP, and the C is on the 5' side. We do have a particular case at n+1 and n-1. In this particular case, because the determinant base is 3' of the SNP, we are therefore looking at the bot strand. If the determinant base had been on the upstream side, 5' of the SNP, it would have been the top strand. So looking at the bot strand, the rule of thumb for assigning genotypes is top strand is alphabetical, bot strand is anti-alphabetical. And so in this particular case, because we're looking at a bot strand, genotype A is T, and genotype B is A. All right? So here are some examples for AT and CG SNPs, and I will actually bring these up so we can take a look. If you look at the first example, you'll see that going n-1 and n+1, we already have an instance. And I'm also showing this in double strand so that you can see how this is consistent for top and bot. So in this case, this strand on the top of it is the top strand because the A is 5', and the other strand going 5' to 3', the T is 3' of the SNP. And so this is the top strand, this is the bot strand, and you can see genotype A is A, genotype B is T here, and for the other strand it's opposite, genotype A is T, genotype B is A. For the second example, we have to actually go out to the second base when we're walking in order to find an instance where the determinant base is on one side and not on the other. In this particular case, this upper strand is the bot strand because the T is on the 3' side of the SNP, and on the other strand, the A is on the 5' strand side of the SNP. So this is bot, this is top, and the genotypes are assigned accordingly. Now here's a case where we've got T's at n-1, but on n-2 and n+2, we've got a T and a C paired up. So this is the bot strand. For this example at the very bottom, we've got A's going all the way across, and we've got T's here, but in the third space, n+3 and -3, the A is matched with a G. And because the A is 5' of the SNP, this one is top. And if you're looking at the other strand, the T is 3' of the SNP, and it's bot. And the genotypes are assigned accordingly. All right? Now, hopefully we have a clear picture of how Illumina's top, bot, and AB genotype standards are assigned. Let's take a look at the Infinium genotyping manifest now, and it will become clear as we go along why this top and bot assignment is important. All right? Infinium genotyping manifests are available in BPM format, which is the format that is used by GenomeStudio. It's a compressed proprietary format. It's also available for customers in CSV format, a format which is a text format that can be opened in Excel if it's small enough, or in a large text editor in Linux if you have a genotyping chip like the Omni 5 with 5 million SNPs that can't be opened in Excel. All the information that is in the BPM file, the compressed version, is also in the CSV file. Now, the CSV version of the manifest also includes probe sequences, whereas the BPM does not. This was a decision that was made early on because we felt that the inclusion of the probe sequences in the BPM would make the GenomeStudio project too unwieldy and too large, And so we decided to exclude them. Of course, nowadays we have such powerful computers that it probably wouldn't have been an issue, but back when we started, computers were not quite as powerful as they are now. But all the information in the BeadChip file can be viewed within GenomeStudio after the project has been created. All the annotations get carried over, everything except the strand sequences will be there, or the probe sequences, excuse me. Now, one of the issues we need to deal with when it comes to the manifests is the naming standards for these manifests. For Infinium genotyping, the BeadChip name and version of the manifest should match exactly the BeadChip name and version on the BeadChip. Now, version changes in the BeadChips represent actual physical differences between the BeadChips. In other words, if you have a version 1 of a BeadChip and a version 1.1 of a BeadChip, they are physically different. We actually create new versions of BeadChips when we run out of the bead pool that was used to create the BeadChips initially. So we have to remake oligos, we sometimes redesign the probes, we have to reattach them to a new set of beads, and then we have to distribute them onto a new set of bead chips. So the manifests are different between versions and should not be cross-matched. If you do cross-match them, you're going to be missing some data because some of the probes don't absolutely carry over from one version to the other, and sometimes they get reassigned to different codes. Manifest revision is a letter designation that's not part of the BeadChip name. Revisions reflect updated annotations and minor updates to the information about the chip, but they don't actually reflect physical changes on the BeadChip. For example, for the Manifest Human Anomaly Express 12 version 1K, the underlying portion of the name is the BeadChip name, and it needs to match the BeadChip name and version of the chips that you scanned. The letter K at the end is the revision letter, which does not need to match the BeadChip. You can use older versions or older revisions of a manifest, but then you won't be reflecting or pulling up the most up-to-date information about the probes and the targets that you can. So we always recommend that you use the most up-to-date version, which is the one that's usually available on the website. Okay. Now let's start looking at the manifest headings themselves. All right. There is a technical note available on the website that describes the manifest headings. It was recently released. It was written by a colleague of mine in the department. So a lot of this work is hers and not mine. A review of the headings, that document lists them alphabetically. I'm going to go through them a little more historically in regard to how the processing is done and how the analysis is done. So the first thing I'm going to look at is the source seq and the source strand. The source seq is the sequence of the SNP and the surrounding area as submitted to our designer software. For RS SNPs, SNPs that are in DB Snip, it is the DB Snip sequence entry as listed in DB Snip. For other databases, it's the same. For the 1000 Genomes Project or for whatever database that our SNPs have been derived from, the sequence is, source seq is the sequence as listed in that particular database. For custom SNP assays, the source seq is the sequence entry as submitted by the customer for design. All right, so again, source seq is the sequence as submitted to the designer. The source strand is the top bot designation of the source seq. All right, so this will be top or bot. It'll be 50-50 because it's, again, top and bot are dependent on the sequence. So it should be a random distribution, and it should be 50-50, half top and half bot. All right. Now, other strand designations, important. The Illumina Strand is the top bot designation of the strand that the probe sequence was ultimately designed on. As I said, when we design these assays, we analyze both the top and the bot strand of whatever sequence we are looking at, and we choose based on the thermodynamic stability, the stability and the appearance of sequences throughout the genome that might hybridize or cross-hybridize with it. So we design on either or, either top or bottom strand. So this Illumina Strand column will tell us which one, the top or the bot, it was designed on. The SNP column in the manifest lists the SNP as reported by the probe. In other words, the alleles that are as based on the Illumina strand, the strand that the probe was designed on. So the SNP column always has the SNP bases as reported by the probes. They also are listed in the column in order of genotype A and genotype B, or rather allele A and allele B. So this SNP is an AG SNP, genotype A is A, genotype B is G. So whatever SNP you're looking at, you can always know which one is A or B by looking at the SNP column. That cannot be said to be true for any of the other columns in the manifest, because those can be derived from the database, and the database may or may not adhere to genotype A, genotype B order. All right. Another column, very important column, is the ref strand. It's at the far right of the manifest, and it's filled with plus and minuses, and it simply tells us what strand, what genomic strand, the plus or reference strand or the minus or the anti-strand, that the SNP and the Illumina strand are on. So this ref strand refers back to the Illumina strand, the design strand of the assay. All right. Another column, top genomic seq. Now, don't be confused. This top is not the forward or plus strand of the genome. This is the Illumina top sequence. So if you look at this column, you'll see the surrounding sequence of the SNP and the SNP in the middle as reported from the top strand, the Illumina top strand. Okay? So all these columns will have some significance, and here's just a review of them again. Now, why is top and bot important? It is because when you run GenomeStudio, you have the option of outputting text reports. And these reports can be designated to report the SNPs in any of several different strand designations. The default designation for output in the Illumina final report is top. If you do nothing else and you just do the default, you'll get top strand alleles. You can also select to report forward strand alleles, which are actually referring to the source strand alleles. Design alleles refer to the Illumina strand alleles. Design, of course, meaning the design strand of the probe. Plus alleles means that you can report this out of GenomeStudio in the alleles that are on the reference strand or the plus strand of the genome. A and B alleles report the alleles as simple A and B, regardless of the strand. Simply report genotype A or genotype B. All right, so you can receive, as a bioinformaticist, for example, any one of a number of these particular reports. And the problem, of course, is what happens if you need a different one. For example, I have received a genotype report that displays the alleles according to X format, whatever format that is. I do not have GenomeStudio or GenomeStudio data. How do I convert this from X to another? The answer is use the top bot designations listed in the manifest. Example 1, I have a top strand report, top allele report. So these are all top alleles, but I want them in forward strand as they would have appeared in DB Snip. What do? Well, you keep in mind what you have and what you want. You have the top alleles and you want the forward alleles. Here we need to look at the source strand. The source strand tells us the top bot designation for the forward alleles, which is what you want. If the source strand designation, the forward, is top, then the top strand alleles you have are already the same as the forward alleles. If the source strand is bot, then you will have to flip the top allele to get the forward alleles. So in this particular example, the source strand is bot in this first one. So for all the alleles in your report, you'll have to flip this one. And for the other one, since it's top already, you don't have to worry about it. All right? Now, example number 2. I have a forward strand report, but I need the alleles listed in genomic forward or plus strand. So you have the forward strand, and you want the plus strand. You will need 3 pieces of information to do this conversion. You'll need the ref strand, which is the designation plus or minus of the Illumina strand. You'll need the Illumina strand, which is the top bot of the Illumina, and you'll need the source strand, which is the top bot, which is also the forward So this gives you the forward top bot designation. Now, for the first SNP listed, the source strand is bot. The Illumina strand is also bot, which means forward strand and the design strand are exactly the same. The ref strand lists the Illumina strand as being minus, so to get the plus allele, you'd have to reverse complement this. Since the source strand or the forward strand is also the same, you would have to flip the forward alleles that you have received in your report. All right. Continuing the same example, but looking at a different SNP, here we have an instance where the source strand and the Illumina strand are actually opposite. Source is bot, Illumina strand is top. So whatever happens, whether you have to flip this one, you won't have to flip the other one. In this particular case, the ref strand says that the Illumina strand is minus. So to get the plus strand, you would have to flip the Illumina strand. Since the source strand is already opposite, we don't have to flip it. So in your forward strand report, which corresponds to source, you don't have to flip this allele to get the plus strand. All right, so you'd have to go through this kind of a transformation, write a script that flips the ones that are opposite and plus, and so forth. The bottom line is you need to compare the top and bot strand assignments for source and Illumina and compare it to the ref strand. All right. So that's a task for all you bioinformaticists out there who have a report that needs to be converted to a different format. All right. All right, that completes our analysis of the manifest for Infinium genotyping. Let's take a look at Infinium methylation manifests. All right. Looking at naming convention, unfortunately, the naming convention for Illumina Infinium manifests for methylation did not follow the conventions like others. Here, we've only ever released one version of the BeadChip. That's version 1. So versioning is not a question with the Methylation 450. We do see that the manifests have been named version 1.1 and version 1.2. Those are the two versions that have been released. But the decimal point actually refers to a revision of the manifest, which means an updating of the content annotations. Actually, the only difference between version 1.1 and 1.2 of the methylation manifest is the inclusion or exclusion of FFPE control probes. Now, we have an FFPE restoration protocol, and we have probes designed to detect whether or not that restoration was done successfully. Now, these probes have always been present on the BeadChip, but in the 0.1 version of the manifest, we did not include the probes in the manifest because we hadn't completed the design of how we were going to use the FFPE restore protocol. And so we released version 0.2, revision 0.2, once we had. So the bottom line is both version 1.1 and 1.2 of the methylation manifest can be used to analyze data. It's just that if you ran the restore protocol, you should use version 1.2. All right. Now, we're going to look at some of the columns that are in the methylation manifest. Here we have the identity of the CPGs in the name column. We have address A and allele A probes, address B and allele B probes. Infinium 1 assays are 2-probe assays, so they have both allele A and allele B probes. For an Infinium 1 assay, the base that gets extended is the base that is after the CPG, so actually the same base gets added to both probes, and we list that same base in the next base column. A's and T's get extended and labeled with red dye, and C's and G's get extended and labeled with green dye, and you can see in the color channel column which dye gets added. Infinium 2 assays are single probe assays, so they only have a probe listed in the allele A area. So you have address A and allele A probes for Infinium 2 assays. You can see in this Infinium column, Infinium type column, which type of assays you have, 1 or 2. All right. Now, the columns of most interest to most people are the columns which list the association of the CPG sites with particular genes, because the main emphasis of methylation analysis is to analyze the effect of methylation state on gene expression. So here we have columns which associate the particular CPGs with particular genes. The UCSG RefGene name lists the symbols of the genes that are being affected. If they're listed more than once, then we're probably looking at alternate splice versions. The RefGene accession column lists the accession number in the, as listed in UCSC and, of course, at the NCBI Ontario website. All right. The RefGene Group is another interesting column. It lists the association of the CPG relative to the gene by location. So if in a particular entry you see TSS 200, we know that the CPG we're looking at is between 0 to 200 bases upstream of the transcriptional start site relative to the direction of the transcription. As you know, transcription takes place in both directions on the genome. So this is specifically relative to the direction of transcription. TSS 1500 indicates that the CPG is between 200 and 1500 bases upstream. 5' UTR, 3' UTR are self-explanatory, and body means specifically between the ATG and the stop codon, irrespective of the presence of introns, exons, transcriptional start site, or promoters. All right? Now, the other columns of interest, because CPGs either exist in islands, which are highly dense clusters of CPG sites, or they're dispersed throughout the genome. Now, so these columns refer to whether or not the CPG is associated with a particular island. Now, the UCSC island name names the CPG island by their coordinates in the genome. Relation to CPG island, we invented some terminology that actually relates the CPG relative to the CPG island that it's associated with. Either it's right in the island, or it's upstream or downstream by 2 kb or 4 kb. If it's within 2 kb, it's a shore CPG. If it's outside to 4 kb, it's a shelf CPG. All right? The other issue that comes up when it comes to designing probes for methylation analysis is the fact that there are Snips distributed throughout the genome, and many of these Snips will end up underlying the probe targets, the probe regions. So here we've actually included a couple of columns that describe the location of these underlying Snips relative to the CPG site and the probe. So in the column probe Snips, we list Snips, RS Snips that are between 10 to 50 bases away from the 3' end of the probe. Probe Snips 10 is a column which lists Snips which are within 10 bases of the CPG at the end of the 3' end of the probe. We do know that Snips that are closer to the 3' end have a much stronger effect on the binding and the assay output. So that's why we make the designation. Now, all of these Snips that are listed in this in these two columns have a minor allele frequency greater than 5%, because at that time we decided we couldn't list every single one of the Snips that are underlying. We felt that 5% was a good cutoff for significance, although that is not an experimentally based number. Now, we did get a lot of requests from customers for a more comprehensive accounting of all the Snips. So we actually created a Snip List that can be imported into GenomeStudio. The Snip List actually lists Snips with a minor allele frequency much less than 5%. It lists the highest minor allele frequency that is listed within the dbSNP entry. So if there are multiple populations analyzed, we list the minor allele frequency of the highest occurring population. All right? And we also, instead of separating them by 10 and 40 bases, we list the exact location relative to the CPG site in the probe. So this list is quite comprehensive. It's updated frequently. It's well described in this technical note that's linked out here. And the list itself can be downloaded from our website on this link. All right? All right. There are other columns in the methylation manifest which were contributed by a consortium of scientists for whom we actually designed this BeadChip initially, the Methylation Consortium. So a lot of this information was contributed by them. We don't have a lot of information outside of what they gave us. So we do have some of the columns listed here, and also some link out to the sources. For example, the Phantom column lists the classifications from the Phantom project as low or high CPG density regions associated with Phantom designated promoters. DMR is the differentially methylated regions, and there's actually a link here to the publication that describes these different types of differentially methylated regions. We also have an enhancer column that was predicted enhancers that come from the ENCODE consortium, and here's a link out to that page. Other columns are not quite as well described: hidden Markov, regulatory feature names, and regulatory feature groups. These were provided by the Methylation Consortium scientists, and we don't have much more information outside of what is listed in the manifest. All right. Now let's look at gene expression manifests. Now, gene expression manifests are relatively simple, so we're not going to have too much information about them. Naming convention, it's the same as for genotyping manifests. You should use the same manifest version to match the version of your BeadChip. The revisions are indicated by a number instead of a letter, and they are preceded by the letter R. So you'll have an R1, R2, R3 for the revisions. As with Infinity Manifest, as long as the version of the manifest matches the version of the BeadChip, GenomeStudio will create a valid analysis project. We do recommend using the most recent available version on the website for the most accurate annotations. All right. As far as gene expression chips go, the important information is really the source of the sequence. So here is a table pulled from the data sheet for gene expression BeadChips available on the website, and it breaks down the different sources for each different expression BeadChip type. You can see here that most of it is from the RefSeq database, and there are different classifications on the RefSeq, but there's also supplementary content that is not as well vetted or as well annotated. So there are several thousand probes here for the human chip, and also for the mouse chip there are different resources. One of the questions we often get, a frequently asked question, what version of the genome build are the coordinates based on? These coordinates are not pulled from a genome build, they're pulled from RefSeq or the various different databases that are available. And so we rely on their information. I have looked at some of the manifests here, and while most of them are from build 37, there are a few probes with build 36 coordinates, and there's no way to separate them out easily. All right, so again, the annotations come from the expression databases and not from the genome build. All right. As I said, gene expression manifests are relatively straightforward and simple. So we associate the intensities from the probes with particular genes, and we use those intensities as representations of the gene expression level. Not much other calculations go on. You can normalize them, and you can, GenomeStudio does have an algorithm for doing differential analysis, but that does not involve anything to do with the manifest. Majority of the manifest content is probe sequence, gene symbols, accession numbers, and annotations from the source databases. Now, many of the genes that are targeted by more than one probe. Various reasons for having different probes. There are alternate splice products from the same gene. There are multiple probes that are targeting the same gene simply for increased confidence in the results. There's a column in the manifest called probe type, and this specifies whether what kind of a probe the probe is relative to the isoforms available for that gene. The letter S in this column indicates that there's only one isoform, and that probe targets that one isoform transcript. I means that there are multiple isoforms available, and this probe targets one specific isoform. The letter A in this column would mean that the targeted gene has multiple isoforms, but that the probe is in a common area for all isoforms. And the letter M is similar to A, in that the probe is targeted to a region that targets multiple but not all known isoforms. So if you're looking for an explanation as to why there are multiple probes targeting the same gene, there's some designation in this column as to why. All right. We're pretty much at the end of the webinar, summarizing this. Manifests are lists of the probes contained in the BeadChip type. GenomeStudio merges the raw intensity output from the scanner with the probes listed in the manifest to output meaningful intensity data and calculations. And for genotyping manifests, the top bot strand designations provide a reference for other types of strand designations and for converting reports. All right. There are lots of resources. Manifests can be pulled from our website. You can either go to the support page and select the downloads button and search for your particular BeadChip. Or if you'd like more information on your particular BeadChip in addition to the manifest, you can click on the BeadChip Kits and Bead Sets link from the support page and get lots of other information in addition to the manifests, which are available there. All right. Technical bulletins. If you have a MyIllumina login, immediately after you log in, the bulletins will be right in your face. This is the page after logging in, and there are the bulletins. You can search for the bulletins. You can view all of them. And there are additional technical bulletins that are covering the methylation and gene expression manifest, and they will be posted to the website